Good afternoon, good morning, good evening, anywhere you are in the world. Welcome. And we have about 333 registrants. That's why we're waiting for some of them to come in. 333 registrants from 46 countries. So this is the first webinar on Gene Drive for 2022. And it's entitled Gene Drive Organisms, There is no one size fits all. So on behalf of the Gene Drive Network and the International Service for the Acquisition of Agri-Biotech Applications, Incorporated, I am Dr. Rodora Romero Aldemida, the Executive Director, and I welcome you to this webinar. So the webinar is, of course, hosted by us, ISA Inc., based in the Philippines. And we gratefully acknowledge our network of biotechnology information centers in Asia and partners in Oceania for their support in this activity. This webinar aims to provide participants with an introduction of different types of gene drives and how guidance and frameworks need to be able to adapt to these differences, to contextualize the need for additional guidance materials on risk assessment within ongoing debates, and provide an overview of the different dimensions of gene drive impact and risk assessments, including environmental and socio-economic aspects. This webinar will run for 1.5 hours. We can extend if there are questions. And we are currently on live streaming at the ISA Facebook page and YouTube channels. All microphones are on mute, so let's put up the rules. All microphones should be on mute, and questions should be entered in the Q&A function only. The chat box will be used by panelists for important messages to the participants. And all the presentations and the Zoom recording will be posted at the ISA Inc. website and freely downloadable by today, later today, or tomorrow. And we also invite you to stay till the end of the webinar and respond to a post-webinar survey, and respondents will be given certificates. So I hope I don't see any more questions on that. So let us now continue formally, and let me introduce our moderator for this webinar. Dr. Guan-Hong Wang is the principal investigator of the Chinese Academy of Sciences. Dr. Guan-Hong Wang is a full professor in the Institute of Zoology, Chinese Academy of Sciences. He obtained his PhD in zoology from Chinese Academy of Sciences in Beijing, and then Beijing, China, and Yale University in New Haven, United States. From 2016 to 2012, he was a postdoc fellow at Harvard University and University of California, San Diego. His lab research focused on animal microbe interactions and genetic technologies for mosquito-borne diseases control. He has published papers in reputable journals, including Cell Host and Microbe Science Advances, Nature Communications, and Trends in Genetics. Ladies and gentlemen, colleagues, let us welcome Dr. Guanhong Wang as our moderator.
Okay, thank you, Dr. Hart's great openings, and thank you all for joining us today in the first webinar of the 2022 Gene Drive webinar series. This is the second Gene Drive series ISAAA organized in partnership with the Outreach Network for Gene Drive Research. Last year, they gathered over 23,000 participants online. Please allow me to introduce our today's distinguished speakers. Firstly, Dr. Alekos Simoni is the scientific manager of Polo, Genomica, Genetica, Biologia in Terni, Italy, which is part of the targeted malaria consortium. Dr. Simoni obtained his PhD at the Queen Mary University of London, and then joined the Imperial College London as a postdoc research associate in the lab of Andrew Chrysanthi, where he contributed to the development of a novel strategy to control malaria, focusing on gene drives. Our second speaker, Dr. Owain Edwards, leads a CSIRO research group in environmental genomics. He was also appointed leader of the environmental and biocontrol domain of CSIRO's future science platform in synthetic biology, where he oversees projects delivering environmental outcomes, including gene drives for biological control and engineering resilience in threatened ecosystems. Before we formally start our discussion today, I would like to invite you to respond to a poll. Here is the question of the day. How many different types of gene drive mechanisms are? So there's 1, 2, 3 or more. I mean, we can see here have lots of multiple choice. Where's the answer? I mean, from a choice, we can see it's really hard to say which is the right answers. I believe Dr. Simoni's talk today will help us to answer these questions. Right, Dr. Simoni? Please feel free to take the floor.
Thank you, Guan, for the nice introduction and for the questions. To hear the full answer, you have to hear my talk, but as I can anticipate that the majority here of the respondents are correct, and there are more than 3 types of gene drives. But let me go through my presentation, and I hope I can help you to understand a bit more about this. So let me share my screen. Okay. I hope you can see my screen. So as one anticipated, my name is Alekos Simoni, and I'm working at the Polo GGB in Italy. And let me just start to brief introduce what is Polo GGB and target malaria, which is one of the main projects that Polo GGB is involved with. So Polo GGB is a company based in Italy, and we provide services and research. We have 3 units. One is focused on medical diagnostics and genomics. The second unit is the genetics and ecology laboratory, which is the one that I am leading. And then we have an immunology lab. As you can see from the images that I put here on this slide, we are working with mosquitoes, and we have a facility which is equipped with a state-of-the-art insectary to study wild type and genetically modified mosquitoes. And we also have large cages where we can test those mosquitoes. Polo GGB is partner of several international research consortia. The biggest one is Target Malaria, and I will talk a little bit about this. But we're also part of other projects funded by the EU, like InfraVec 2, Isidore Scenarios, and also we were part of one consortium funded by the DARPA. Target Malaria is a not-for-profit research consortium that includes scientists like myself. There are protein engineers, molecular biologists, medical entomologists, population biologists, and so on. We have also a great number of advisors in the field of risk, regulatory, and community engagement. And we operate with teams in Africa, Europe, and North America. And the mission of Target Malaria is to develop and share new cost-effective and sustainable genetic technologies to modify mosquitoes and reduce malaria transmission. And we are focusing, therefore, on modifying mosquitoes for malaria control, and the technology that Target Malaria is developing is gene drives. So let me start discussing about gene drives, first with the definition of gene drives. And for this, I take the help of this very nice publication that has been published about 2 years ago in the Journal of Proceedings National Academy of Science, where the authors try to standardize the definition of gene drives, because it's very important to have a common ground in order to discuss a topic. And the authors here propose the definition of gene drives based on 3 elements. So the first definition of gene drives, which is the most common, is define a gene drive as a process, is a biological, a genetic process, a phenomenon of biased inheritance, whether a gene or a genetic element within the genome of an organism, it is inherited more than what you would expect from Mendelian inheritance over the rest of the genomes. And that leads to an increase in frequency of this element over time, even if this gene or genetic element can pose some fitness cost. So the first definition of gene drive, it's a process. A gene drive is a process of biased inheritance, that a gene that can increase in frequency over time. But for gene drive, we don't only mean the process of biased inheritance, but we also mean the construct, the molecular components that lead to this process of biased inheritance. And those 2 terms sometimes are interchangeable. And for gene drives, sometimes we also need the tool to achieve a specific goal. For instance, we can talk about the gene drive mosquitoes in the sense that those mosquitoes contains a gene drive, and those gene drive mosquitoes can be a tool, for instance, to change a population to control malaria, for instance. So let me try to explain a bit more what gene drive is in the context of a population. So imagine that you have a modification which is not a gene drive, which is here represented as green mosquitoes. By the way, you will see a lot of mosquitoes on my slide because I'm working on mosquitoes, but this concept can be applied also to any other organisms. So imagine you have a modification which is not a gene drive in a population, and imagine that this modification is present, like exemplified in this, at 20% in your population. Since any modification follows Mendelian rules of inheritance, by which only half of a progeny have a chance to inherit this modification, because you get a copy of your genes from the father and 1 copy from your mother, this modification is inherited to only half of the progeny, so on after generation. That means, in terms of frequency of this modification, that the frequency of this modification stays the same, remain constant over time. If you have a gene drive instead, and you have this gene drive, let's say at the same 20% frequency in the population, because gene drive can bias its own inheritance, this modification is inherited to all of the progeny, generation after generation. And as a consequence, this gene drive can spread through the population and increase in frequency. And this can happen even if the population, even if the gene drive can impose a fitness cost. So that's the main concept of a gene drive. It's a type of modification that spread over time and increases in frequency. And already here we can start to define 2 main type of applications for gene drive, which are suppression and replacement. And they are similar in one way, but the outcome is very different. So the first type of application for gene drive is what we call population suppression, where you introduce a modified organism in a population, indicated here in green, and this gene drive organism spread over time, and while they do so, they reduce the population size to a point where you have a much smaller population. So you suppress the population. You can do that, for instance, targeting fertility or other aspects of the organisms. This is a very efficient way if you want to control a nefarious organism like a vector for disease or an invasive species. The other approach for gene drive is what we call population replacement, where you introduced a modified organism in the population, this spread through the population and replace your old population. So the population size remain the same, but at the end, you end up with a different type of population carrying your modification. And if you want to apply this, for instance, for malaria control, you can spread a mosquito that is refractory to the disease, for instance. So you replace your population with a variant which is beneficial. So gene drive can be used for both approaches because the gene drive is this process that allows a modification to spread through the population in a very efficient way. And I'm talking here about mosquitoes, but gene drives can be applied to many, many different organisms. It can be applied for different types of mosquitoes. It can be applied for crop pests like Drosophila or medflies. It can also be applied to mammals. For instance, there are proposals to apply gene drives to control rodents, which are invasive in several parts of the world. But how then a gene drive, this process of biased inheritance, can have an outcome which is useful in terms of population suppression or population replacement? And you can do that in two ways. One way is to include this drive, to insert the drive inside an essential gene, and therefore to disrupt the function of the gene, and therefore making a mutation. And this mutation can also, for instance, can target the fertility of the individual or be a lethal mutation, and therefore you have a phenotype due by the fact that the gene drive destroys an essential gene. Or you can link your gene drive with an effector as a form of a cargo that is driven together with the gene drive while the gene drive spreads. So you can use the gene drive to disrupt a gene or to link another gene, an effector, with the gene drive and allow it to spread together. Now let's go through the different types of gene drives. The most common one is what we called homing-based gene drive, which is based on the homing process, which is a natural phenomenon that can occur in the cell, which is based on DNA cleavage and repair. And this system can be engineered for the scope of making a gene drive. So how it works? It works by inserting a nucleases. So the gene drive in this case is based on a nuclease, which is a DNA cutting enzyme. Nowadays, the most common nuclease that is used in biology is CRISPR because of its efficiency and ease of use, but is not the only nuclease that can be used. So in this case, the gene drive is a nuclease that when it is expressed, it recognizes a specific site in the genome, and the gene drive is inserted within its own recognition site on the homologous chromosome of the organism. Therefore, when the gene drive is expressed, it cleaves the DNA, induces a DNA cleavage, a break, and this break has to be repaired by the cell. And if the cell uses the cell repair machinery, using homologous recombination, using the chromosome contain the gene drive as a template for repair, it can copy over the gene drive from one chromosome to another. And this is the process of DNA cleavage and repair that leads to increasing frequency of the gene drive because of this DNA repair. This process is called homing and allows us to increase in frequency of the gene drive over time. This has been proposed many years ago as a tool for controlling population if the gene drive recognizes a site within an essential gene. And while the gene drive can spread over time, it can reduce the population fitness because it imposes a load on the population. So these type of gene drives can be built using any type of nucleases. Of course, nowadays we use CRISPR because it is the most efficient, but it's not the only one. And this type of gene drive can be used either for population suppression, if you target with your gene drive an essential gene, or for population replacement, if you link to the gene drive a cargo. This has been developed for mosquito control with very promising results in the lab. And this is considered a low threshold drive. So the threshold for gene drives is the concept that some of the gene drives need to reach a certain frequency in the population before they start to spread. Some others gene drive, like this one, can spread even from a very, very low initial frequency because of this process of biased inheritance. So this type of gene drives is considered a low threshold drive because it can spread from a very, very low number of initial individuals, theoretically, even from very, very few. And also another concept about the gene drives is the ability to spread. And this can be limited in time or space. And this type of gene drives, based on homing, they are considered to spread in an unlimited way. So that is not limited over time or over space. Of course, the spread is dependent on the fitness of the organism, and there could be ways to restrict somehow the spread using genetic characteristics, but maybe we can discuss that. There are some specific questions. I just want to highlight here a couple of publications from the lab where I'm working, where we show that a type of gene drive like this one, targeting female fertility of mosquitoes, led to population collapse in the lab. And this was shown both using small cages population, where we showed that the gene drive could spread, reach 100% frequency, and leading to collapse of the population. You see here those are the X output from the population. And that was also shown in large cages in the lab where I'm working, where the gene drive spread also there to fixation, 100% frequency in different release scenarios, and leading also there to population suppression. So this is shown to be very effective in the lab to control mosquito populations. Another type of gene drive is not based on homing, so it doesn't require cleavage of DNA and homologous recombination repair, but is based on the idea that if you want to control a population, you can change the balance between the sexes. And if you switch the sexes toward males only, for instance, you can reduce the population size. And the reason why the idea is to make a population male only is because for mosquitoes, for instance, it's only the females that bite and can transmit diseases. So if you remove the number of females, you reduce malaria and disease transmission. And also because the population size, it's usually dependent on the number of females rather than the number of males. So if you have a way to bias the population towards males, you can induce population control and also reach population suppression. So imagine that you have an effector of some enzyme that is active on the male gametogenesis. And what it does, it targets the X chromosome in an organism with heterochromatic chromosomes. If it does so, you can eliminate all the sperm with the X chromosome, leaving only a Y-bearing sperm surviving. And this, after fertilization, can generate only male progeny. So that's the idea. If you are able to link this effector, this enzyme that destroys the X chromosome, to the Y chromosome, all the progeny will inherit it. And therefore, this can bias in the population because you will generate males-only progeny, and all those males will carry this effector, and therefore, generation after generation, this contact will spread to the population and will eventually collapse. This also is considered a low threshold drive, and also this can spread in an unlimited way, depending on the fitness and the ability to bias the sex toward males. For mosquitoes, this has not been achieved yet because there are some biological limitations to express effectors from the Y chromosomes, but there are some alternative ways that can achieve a very similar result. So this is also a very promising type of gene drives. Another type of gene drives I would like to describe to you today is what we call a split drives. And the split drive is similar to the homing-based gene drives, but it is constructed in a way that the components of the drive elements are not linked together, but they are split in different chromosomes. This can be easily done with CRISPR because, as probably you know, CRISPR is based on two components. One is the Cas9, which is the protein that makes the cleavage to the DNA, and the other component is the guide RNA, which is the component that allows us, the Cas9, to be specific, recognize a sequence in the target gene to make the cut. And you can split those two components into different chromosomes. And if you do so, they will not be inherited together unless they are present in the same organism together. So when they are together, for instance, if you generate two strains, one with the guide RNA and one with the Cas9, when they are coming together, the Cas9 acts on the guide RNA, and the guide RNA recognizes the target site, and only one of the two components is homed. So only one of the two components is inherited all the time, while the other component is inherited in a Mendelian fashion, and therefore they don't increase over time both components, but only one of the two. As a consequence, you will have a spread of those elements which follows a pattern like this one. So they start to increase only for a limited period of time, and then they start to decline because the two components are not linked together, but they are splitted. And this is a type of gene drive that can have several applications if you want to constrain in a time fashion or in a space fashion your spread of the gene drive. So it can be more efficient, for instance, if you want to target only a constrained population, or if you want to have the effect of the gene drive limited only for a short period of time. It is less efficient if you want to target a wide, large population. The last type of gene drive that I would like to discuss today is what has been called MEDIA. That stands for maternal effect dominant embryonic arrest. And this is a different concept, completely different compared to the one that I described before, which this is not based on DNA cleavage, but this is based on a toxin, which is expressed from a maternal promoter and a zygotic antidote. So, and the way that this can spread is because it causes the death of the individuals that doesn't carry the construct itself. So you have a toxin which is it kills all the individuals that doesn't have the antidote. This also is more challenging to construct because it requires different components and are also more difficult to transfer to other species because the toxin and the antidote might work in a very different way in different species. And this type of drives, it's usually threshold dependent because it requires the construct to be present above a threshold in the population before it starts to drive, similar to the split drive, which is also threshold dependent. This has been shown quite effective to work in the lab from people like the lab from Omar Kabari and others, and has been proposed for population replacement approaches in Drosophila, especially Drosophila Suzuki. So this also is a threshold dependent drives. So I hope I can help you to understand that there are many types of gene drives, and many are exist in nature and many are engineered, and they can be classified according to different characteristics. The first one is the molecular components of the drive. They can be nucleases, CRISPR, or can be antidote or toxins or other mechanisms. And the main difference between the drives is the aim of intervention. So you can either develop a drive for population suppression or for population replacement to drive parasite refractoriness, for instance. And the intended effect of the drive in the organism also can be very different from application to application. You can induce sterility, you can target the mating ability of the organism or the ability to feed, or several behavioral traits like the ability to fly for mosquitoes, for instance. The time of action also within the organism can vary between the different types of drives. They can be restricted to the germline, they can be post-gametic or active only in the adult. And the spatial scale of the gene drive also can be either localized or not localized, or act only or some specific set of allelic variation within a population. And I would like to finish with some consideration in general about gene drives and some of the challenges that are facing the field of gene drives to move the technology from the lab to the field. The first consideration is about the efficacy of the gene drive. And of course, since gene drives are based on biased inheritance, the ability to bias this inheritance is what makes them more efficient, and different type of drives have different efficacy in that respect. But there is also another important aspect to determine the efficacy of the drive is the fitness cost of the individual, because any type of genetically modification usually impose some cost, and if the cost is too high, the drive cannot spread. Then, of course, the efficacy of the cargo for population replacement, that's what also lead to the efficacy of the strategy, and therefore also how they are released, if they require a threshold or not, and the logistic of release. Something to consider about the persistence, which is how long the gene drive stays in the population, and that also depends on the type of application. If the application is a population suppression, potentially the gene drive will disappear when the population decline, while if you are developing a gene drive for population replacement, the persistence is potentially indefinite because you want the modified population to stay there forever. So that also poses different questions in terms of policy. And also to consider that persistence is not equal to spread. You can have a very quick spread, but then the persistence can be very low. So there are two different concepts. And also the localization of the gene drive also depends on the application and the scope of interventions. And therefore also, for instance, if you want to target malaria control, reducing the number of mosquitoes, you want to have a strategy which is active on a very large scale and persist for a long time. While if you want to target a more urban organism, or if you want to target, for instance, a pest for some crops that affecting only some fields, but you want to limit the spread, so you may need a gene drive which is maybe less efficient, but can be contained easier. One of the main limitations for gene drives is resistance. Resistance can arise in different ways, and it can be developed in the population to stop the drive. And there are strategies to mitigate resistance. One is, for instance, the choice of a very conserved target site, or multiplexing, so using multiple target sites for the gene drives, or controlling all the components in a very precise way. So there are ways to mitigate resistance, but it's still one of the main challenges probably for gene drive in very long term. Then, of course, there are challenges for the policymakers and the regulators, because gene drive is a novel technology. It has not been used yet in the field. And as I explained to you, since there are so many different applications and types of gene drives, the risk-benefit analysis must be done on a case-by-case basis. And sometimes this would also require a supranational or regional regulations, because the fact that gene drive may act on very wide area. And then not to forget that the gene drive must be also accepted by the public, and there must be therefore a stakeholder engagement approach that take in consideration many aspects, the novelty of the approaches, to deal with the misinformation that sometimes happens around topics which are extremely complex. But I also would like to point out that gene drive so far has been also a very good example of responsible science, where all the people involved in developing gene drives has been the first one to propose a recommendation and safeguards guidelines for the use, for the correct and ethical and sustainable use of gene drives. This, I think, is a very positive aspect. With this, I finish my presentation. I thank you for your attention, and I'm very happy to take any questions at the end of Owain's presentation. Thank you.
Okay, thank you, Dr. Simoni. Thank you for your wonderful talk. Thank you for this clear explanation of the different types of gene drive mechanisms. This is very important to understand how complex these organisms are when formulating policies. Now, I would like Dr. Owain Edwards to help situate gene drives in a broader policy debate. Dr. Edwards, how gene drives guidance and frameworks are integrating all these differences. Let's welcome Dr. Edwards.
Thank you. I'll just share my screen. Well, thank you for the introduction, Guan, and thanks for the outreach network for gene drive research and for ISA for the opportunity to speak to you all today. As Guan said, my name is Owain Edwards. I work for CSIRO, which is the National Scientific Research Organization in Australia. And I have a leadership role in what's shown here, which is the Synthetic Biology Future Science Platform. And that includes oversight of our gene drive research. And our gene drive research is mostly focused on invasive species, unlike what Alekos was talking about. And so we're targeting more vertebrates, such as rodents and invasive fish, rather than mosquitoes. But I won't be talking about that today. I'll be talking about regulatory and policy considerations when we consider the diversity of gene drives. And as you probably know, precedents are a critical guide for regulatory policy, and the best precedent for synthetic gene drives of the kind that Alekos has been describing use a natural gene drive system, which uses Wolbachia symbiotic bacteria. And this has been used in Australia already for the control of dengue mosquitoes. And I have been involved in obtaining the necessary permits for Wolbachia mosquito releases, and also in preparing risk assessments for these releases. And so I've listed here some of the learnings from the regulatory process for Wolbachia. And one of the key learnings is that the existing broad generic risk assessment frameworks that exist for other technologies, in particular control technologies, are suitable for new approaches like gene drives, as the questions that need to be addressed are common amongst all of the control technologies. So we shouldn't be looking to design a specific regulatory framework for gene drives, because as we've seen from Alekos's talk, there's much diversity, and any framework that's designed for one gene drive probably will not work for another type of gene drive. The second point that I want to make here is that we should be taking advice from all available relevant guidelines, whether these be for gene drives or for similar technologies. And thirdly, it is critical to engage with all stakeholder groups, ranging from experts to the general public, to identify and evaluate all perceived risks. So one of the published guidelines available is the WHO guidance framework for testing genetically modified mosquitoes, which has recently been updated to include gene drive approaches. And so these guys, importantly, recommend a phased testing pathway in which new genetically modified mosquito strategies move from the laboratory to confined environments and eventually to field releases, and each transition is dependent upon satisfactory demonstration of efficacy and safety. And now the original phased approach is shown here, and then has been updated for gene drives and includes a recommendation, obviously, for physically confined field studies. Now, the Gene Drive on the Horizons publication from the US National Academies provides additional information that can be added or considered as part of the WHO advice. And that's what I'll do on this slide. So one important result from both the population cage studies and the early small-scale field trials is to validate the models that form an integral part of the risk assessment process for gene drives. Unlike some of the earlier technologies, it is absolutely recognized that modeling is going to be a critical component of risk analysis for gene drives. Now, it is also possible to use ecological or genetic containment when it is not possible for some organisms. It may be the case that you can't do physical containment, but obviously physical containment is always preferred. And finally, for the field trials, it is important to consider starting with less risky genetic constructs, such as a non-driving form of a genetic modification. And this is actually the step that Target Malaria is taking in the first instance. Or using a drive that doesn't have a cargo or doesn't disrupt a gene, depending on how the drive is designed, so that you can look at the drive, the dynamics of the drive through the population without it actually impacting on the fitness of the target organisms. For those low threshold drives, you can consider actually starting those drives as a split drive approach. And Alekos talked about the split drive and the fact that it stays for a shorter period of time. So the spread is limited in time in the target population. And so this can be done as a less risky option than putting out a low threshold drive in the first instance. And finally, there are a number of genetic localization strategies that are being developed by a number of different research groups around the world. And if these aren't appropriate for the final drive, because you want it not to be self-limiting, then they can be considered as part of the test drives that you do as part of a staged process of release. Now, I also want to talk about this document, which is put out by the Food and Agriculture Organization, the FAO of the UN, which is called the Pest Risk Analysis for Quarantine Pests. I see this as being relevant in the context that we are releasing a new organism into the environment that we want to ensure does not become a pest in its own right. And of these stages of pest analysis that are listed in the document, the first one probably isn't relevant because I think the probability of introduction is we're introducing on purpose for a gene drive. But the other three I think are relevant. And I want to speak specifically about the fourth point here, which is pest risk management. And this is basically describes or assesses how likely we can manage the pest using conventional methods if it were ever to become established and turn into a pest. I think that's an important consideration as part of the regulatory and policy system. There's also an annex to this document, which does address and develop pest risk analyses for living modified organisms or genetically modified organisms. And this annex recommends considering risks relative to the unmodified organism. And those risks should be considered both in terms of direct effects, so the altered characteristics of that organism which might make it into a pest, and the indirect effects. And this is, indirect effects would be the effects on other species within the ecosystem due to the changes, not only in the quality, but of the prevalence of the target, which could be the reduction in the target, of course. Before proceeding, I do want to emphasize again the importance of the stakeholder consultation process, including the general public. And I did mention earlier that this was done for the Wolbachia releases that we did in Australia, and recently a similar approach using a quantitative risk framework, which is again what we use for Wolbachia, was published from the target malaria team for the gene drive approach. I think this is great. I'm not going to go into this in detail because the next, I think the next presentation in this series will be talking about risk assessment considerations in detail. But finally, I do want to talk about an additional step that needs to be considered for gene drive organisms, and this was alluded to in Alekos at the end of his talk, that usually is not considered for other technology releases, and that is the probability of spread after establishment into other regulatory jurisdictions. And this is especially important for those gene drives that are population replacement gene drives, where we expect the persistence to be very high, because the longer the persistence, the more likely it is that that drive has the opportunity to spread into other jurisdictions. This is an important point that's been brought up as part of discussions at the CBD, the Convention on Biological Diversity meetings, and is addressed, or can be, the issue can be addressed as part of the Cartagena Protocol on Biosafety, which does offer a framework to evaluate impacts and also potential penalties. So if we go back to this from the WHO document, we do see at the end post-implementation surveillance. And that absolutely will be critical in order to guide as to whether we have to consider the movement or whether that movement into other jurisdictions is being observed. But we think it is absolutely important to start as much as we can to answer questions about whether this is likely to happen and where it is most likely to happen prior to the release of the gene drives. And this can be done through modeling, and it can be done, and that movement can occur because of natural movement, wind-borne movement, for instance, and also human-assisted movement, such as movement on transport vehicles. In terms of natural movement, at CSIRO, we've developed a tool called Tapas, tool for assessing pest and pathogen aerial spread is what it stands for. And this can be used to model wind-assisted long-distance movement of insects or spores. And this is based on modeling by NASA in the US that was originally used to predict the spread of nuclear fallout and is still being used to model the spread of ash clouds from mosquitoes. And we have applied this. This is for a virus vector insect, looking at the movement both from, this is north of Australia into Indonesia, from Indonesian islands into Australia, or from Australian islands into our neighbors and into Indonesia. We are also looking at shipping lanes, which are known to be important for the spread of dengue mosquitoes, as they tend to hitchhike or can hitchhike onto these vessels. Now, these movements can be predicted through a modeling analysis of the shipping lanes, and they can be tested using genetic methods. So we are doing this, and we are also using similar approaches with airports. So testing at airports and mosquitoes that may come in this case, through airplanes. And finally, again, using genetic approaches, we can look at historical incursions as a means of predicting what might happen in the future, as it can identify pathways of spread of particular organisms. Here, we're looking at fruit flies, and fruit flies, in fact, are a potential or a proposed target for gene drive control. In this case, we identified that an incursion of fruit flies into New Zealand in 2014 came from the east coast of Australia, whereas in 2018, an incursion came instead from New Caledonia. So here we've identified two pathways, historical pathways of movement, either from natural movement or through human-assisted movement, but a pathway that definitely exists for fruit flies for movement between jurisdictions. So I want to finish just by giving these take-home messages. The first one is to cover the diversity of gene drives. We should use an established generic case-by-case regulatory assessment approach. And this is also what Alekos said as part of his talk. We should take advice, excuse me, by published guidelines, whether they're focused on gene drives themselves or other types of control technologies, and by engaging all types of stakeholders. The field releases, I believe, should be staged not just by scale, but also by technology, moving from less risky to high risky technologies as we go through the stage process. And the likelihood of spread between jurisdictions after establishment should be examined in depth for gene drive organisms, which is unlike what is normally done as part of the regulatory process, because the regulators basically just focus on their own jurisdictions. So thank you very much for your attention. I'll be happy to answer questions as part of the Q&A process.
Okay, thank you, Dr. Edwards. Thank you for a great talk. Thank you for providing us with an overview of what policy and regular tools are needed to manage gene drive research and potential use. Now, it's time for the Q&A session. I would like to invite the audience to use the Q&A box to send you questions if you haven't done it yet. I see here some questions for Dr. Simoni already. So let's see. Okay, here are lots of questions. Okay, so the first one is from Covid Guja. Is DL gene a case of gene drive?
I'm not sure what DL stands for. I guess it stands for dominant lethal. In that case, it depends, not strictly, but it's not very clear. So maybe if Govin would like to clarify better the questions, I can give a better answer.
Yeah, I mean, I agree with you. I'm not sure the DL exactly, but I guess you are right. So let's maybe move for the next question and then we go back, okay? So this one is from the Sanghanika Chanda. In case of a homing type of gene drive, what will be guide RNA sequence? How are we going to introduce gene drive construct in one chromosome firstly?
Yeah, this is a good question. So the guide RNA sequence would be the target gene that you want to introduce into target, and that's going to be the integration site of the gene drive itself. And the gene drive can be introduced in one chromosome by CRISPR, for instance, or by other means of genetic manipulation. There are several gene editing technologies that allow us to introduce a construct into the genome. Homologous recombination mediated, for instance, based on CRISPR, it's one of the approaches that we are using, for instance.
Mm-hmm. Yeah.
Okay, perfect. Okay, there's another question from Jackson Chamber. Most other engineered gene drives are similar to other natural drives as well. Many different mechanisms are found in nature. This includes the natural gene drive in multicellular animals. So
I think this is more a comment than a question, probably referring to one of the previous questions.
This is maybe some comments from Jackson. I mean, okay.
Yes. Thank you, Jackson.
Yeah, but that's really good comments. I mean, so, and there's one question from Mubin Aslam. Can we use gene drive for crop improvement?
So in principle, I think can be used. At the moment, gene drives are mostly developed for tackling important problems like disease or for controlling invasive species. So that's what drives at the moment the field of gene drives. But gene drives can be applied to any organism which are sexually reproduced, and they used to have, in order to be efficient, a short reproduction time and need to produce a number of progeny. So in theory, it could be used, but there are, to my knowledge, not many applications for crop improvement at the moment using gene drives.
May I comment on this?
Yeah, sure.
So, I mean, it could do, but it would not make sense to do so because in crop breeding, you have full control over the breeding process anyway, and using a gene drive would be an extremely inefficient method of getting inheritance of a particular trait when you have full control of the breeding process anyway. It would be much more efficient just to use conventional genetic modification methods.
Okay, thank you. So go back to the first question. Yes, the DL means the dominant lethal gene. So is the DL gene a case of gene drive for Dr. Alekos?
So not strictly, because a dominant lethal would not allow it to spread because it's dominant lethal. So gene drives usually target recessive characteristics that can spread. Dominant lethal are considered an SIT, for instance, approach where you need an inundative release of dominant lethal to suppress a population. So in that respect, is not considered a gene drive.
Great.
I would say though that there are suggestions of a combined approach of using a dominant lethal that doesn't express itself until several generations. So you allow a drive to drive itself through the population, and then you trigger a dominant lethal effect. And so that overcomes part of the problem with the dominant lethal approach of having to have such a high frequency in the population, which you usually have to release very, very large numbers. So if you were able to drive the system through the population first and then trigger the dominant lethal effect, then you could actually have a very effective drive that would also be self-limiting once it was triggered.
Okay, perfect. Okay, here is one question I think for Dr. Edwards. How would one apply any type of the gene drive technique at a commercial level to control the vector population in the environment?
Yes. So, I mean, I don't think any of us that are working I'm not aware of, at least certainly we are not interested in gene drives. We're interested in them for public good rather than for commercial purposes. I think since we're looking for more of a permanent or a long-standing response, in general, gene drives are just like most biological controls, do not have a very good business plan. You would have to engineer the drive in such a way as it would have be very effective but very self-limiting in order to need to go back often to a commercial entity in order to purchase it. I don't think that we have the engineering capacity yet to be able to do that. I don't know what Alekos would say to that question.
Yeah, okay. I agree with you.
Okay. So is it possible to do the same gene drive with the bacteria?
So because the gene drives that we have that are at least the designs that we use at the moment are inherited through sexual reproduction. For most bacterial targets, it would be very difficult to design a very efficient gene drive that was inherited in that way. There have been at least one proposed drive mechanism that I've seen that purports to be functional within a bacterial system, but I wasn't very convinced by what was published, and there isn't very strong evidence for efficacy that I've seen.
There's one comment from Dr. Hector Quimada about the commercial interest. I'd just like to jump in here. So regarding commercial interests, companies would also probably not be comfortable releasing something that they couldn't control post-release for liability reasons. Do you agree on that? Dr. Alekos and Owain?
I think I agree on that. Yes.
Okay.
That's a very good point.
Yeah. Okay. Let's move on to other questions. There are some interesting ones, especially from Nahid. Okay, this is from Nahid Kauser. This is for Dr. Alekos. The gene drive with cargo has how much success rate? Is it successful? Is it possible that incorporating gene knockout after few generations, what would be the effect of biased inheritance on environment?
Okay, thank you for the question. It's very difficult to give a general answer to this because it will be related to any type of specific intervention and gene drive construct. In the efficacy of the gene drive with a cargo depends on two aspects. The first one is the ability of the drive to drive, so the biased inheritance, and that can vary by organism and by different approaches. And then the second component is the efficacy of the cargo to make the desired effect. If you are, for instance, making a mosquito refractory to malaria, you need to have a drive that can spread through the population. And then you have the cargo that is efficient in preventing the mosquito to transmit the parasite. And therefore, those two are independent to each other, and they have different efficacies. So the success rate of the application depends on the ability of both components to be effective, and that it depends on the different types. Regarding the possibility that the cargo get unlinked from the gene drive is not impossible. It's something that has been proposed. It could happen through either mutation, that it just become inactive and then you end up with the drive without any effect, or it can be transposed in the genome. So there is possibility that the cargo or the gene drive gets silenced somehow. In terms of the effect in the environment, that also depends on the target species. For a general consideration for gene drives is that since those processes that we described today rely on mating, therefore only species that can mate to each other are targeted by the approach. So usually gene drives are very species specific, which is usually much more specific than other interventions trying to solve the same problems like insecticides or other interventions. So in terms of the impact on the environment, it can be assumed to be low in general, but that also depends on the target species. If you're targeting also invasive species, usually those are not really beneficial to the environment. So therefore, that's also a less important problem in the sense that you're already targeting an invasive species. But the role of the species in the ecosystem is also something that, for instance, we at Target Malaria are taking very seriously. And there are several parts of the project that are looking at the role of the Anopheles species that we are targeting within the food chain and the whole ecological niche. So that's something that we are looking actively.
Thank you, Dr. Simoni. And I think here I have another question, is that what could be the consequence of population suppression and replacement using gene drives as far as food chain is concerned? Would the future environment becomes all unnatural because this approach will eradicate the original population or wild types?
Okay, thank you. I think I partly responded to this on the previous questions about the ability of using gene drives in a very species-specific manner that can be targeted to the nefarious species. For instance, to target malaria, we can specifically target the mosquito species that transmit malaria to human and not all the mosquitoes. And sometimes we also underestimate the complexity and the number of different species in the environment. For instance, specifically for malaria, there are more than 3,000 species of mosquitoes in the world, but only very few are responsible for transmitting malaria. And therefore, we could target the few mosquito species which are responsible for malaria transmission without impacting the different mosquito species which are present in the environment. I don't know if Owain would like to comment a bit more on this question.
No, the only thing I would add is that that's part of the risk assessment process. That has to be. I mean, it's an important part of the risk assessment process that the impact on the ecosystem as a whole has to be evaluated. And I agree with what Alekos has said.
Yeah. So, okay, so there's one question I think for Edwards, Dr. Edwards. How is the Oxitec technology currently being field tested, similar or different from gene drive system discussed by Dr. Simoni? I think this is from the Florida, right now.
Yeah. So the Oxitec technology is the same as the dominant lethal that was the previous question. It's an example of a dominant lethal system, and so the answer we gave before about dominant lethal would be the same as for this one. It's not a gene drive system. It is a system that relies on large numbers of being released, high frequency of the dominant lethal in the population for a very short time, as opposed to gene drives, which is small releases that build in frequency and are persistent over a longer period of time.
Okay. Yeah, I think right now there are no allowed for the gene drive for the field trial in the whole world, right?
So there are no synthetic gene drives or engineered gene drives for which there is approval for doing releases at this stage. No.
Okay. There's one more question for Owain. You point out the need for considering transboundary movement post-establishment seems to point to a regional regulatory system. Are there precedents for that type of system that we can use for setting something up for gene drives?
So I did mention the Cartagena protocol. That's pretty much the only policy framework that exists for the assessment of this, and there aren't, and you have to be signatory to it. And there aren't as many countries that are signatories to the Cartagena as to the CBD in general. But again, I would point out something that Alekos referred to earlier, and that is that the gene drive community is being quite active and proactive in terms of addressing these issues. And so certainly with our community, we are actually we are bringing together regulators from different jurisdictions to specifically address this question because of the absence of an existing framework. So I think we'll be pushing this ahead. The community will be pushing this where it doesn't exist sufficiently.
Okay, perfect. There's one question from Sridhar. Can the gene drive will be productive in managing invasive waste with species in farming systems?
I can do this if you want, Alekos, because we are interested in gene drives against weeds. The designs that currently are being used for insects and for animals do not seem to function well in plants. And there are other important considerations in plants, such as seed banks are very are very difficult to overcome when designing a gene drive, because you always have the wild type material that continues to appear every year. So there are challenges with gene drives for weed species. It's also very difficult for tetraploids, the hexaploid, etc. The high ploidy levels in plants that we sometimes see make them difficult targets for gene drives as well. And so there are challenges, but I do expect that eventually we will see gene drives for weed species.
Okay. So there's I think there's one more question. I think is that aside from insect vectors like the mosquitoes, what other organisms are currently used in research on gene drive?
So I can take this one. So yes, gene drives has also been developed to target not only mosquitoes, but also insect pests of crops, like Drosophila suzuki, for instance, or the medfly. So there are other examples where gene drive can be applied. In general, if you have an organism where you know the genome, you are able to genetically manipulate, it's sexually reproduced, it has a short reproduction time, it can be amenable to be controlled by a gene drive.
Okay, I think here I have another similar question. If a gene drive could be applied to mosquito causing a malaria, could it also be used in species that cause pathogenetic effects to crops? Example in rice. What's then the possible approach?
I think the approach could be the same. So you could either use it for suppressing the population that cause the transmitted disease to plants, or you could also use it to drive an anti-pathogen cargo to the species if it is possible to manipulate those organisms. So I think that the ability to manipulate those insects can be the limiting factors, because for some species, there's something that is well achieved. For others, it can be much more complex. And also very important to consider is the generation time, because gene drives are very effective in organisms that have a shorter reproduction time. So mosquitoes reproduce every couple of weeks, like flies, even mouse, for instance, or rodents can have generation time of a few weeks. But if you're targeting, for instance, some insect species that have a reproduction time of 1 year, you will see the effect after 20, 30 generations that can be way too long for the intervention to be efficient.
Hi, Dr. Owain. For this question, like for the plant gene drive, is there some undergoing field trials or no?
No. There, as far as I know, there is not a functional gene drive in plants. There's been one publication of what was sort of a gene drive, but very low efficiency in Arabidopsis. But I haven't seen any indication of an effective gene drive in plants. And so certainly there aren't field trials for gene drives in anything at the moment for synthetic gene drives.
Okay.
Dr. Guan, there's one question from Zaira. Can you read it? I posted it at the chat box.
Yeah. Okay, in the chat. Zaira, I didn't see where's the Zaira question. Okay. Oh, here.
Okay.
Oh, here. Sorry, here. Yeah, this is from Zaira. For both speakers, countries are currently discussing the need for additional regulation on gene drive at the UN Convention on Biological Diversity. Given the complexity of gene drive mechanisms and gene drive mechanisms, do you think this is the right forum for what?
For that.
What would you consider a good outcome?
Owain, do you want to go first?
Yes, well, we actually did we had a in Australia at least, we held a workshop and a conference brought researchers and regulators together to ask the specific question as to whether the regulatory processes that currently exist had any gaps or would be appropriate and effective for regulating gene drives. And the unanimous opinion that came out of that was that the existing regulatory processes would be just as effective. And as I said, if you cannot design something that will be specific for gene drives because the gene drives are so varied. And we now have new gene drives, components of which are some of it is natural and some of which is GM. And I know there's an interest in focusing on the GM nature of gene drives. Well, we should be looking more at the dynamics of drives themselves and not worry about whether they're GM or not, because it's the important factors to look at are how they behave in the environment.
Yes, I just want to add that I completely agree that there are so many different types of gene drives and different applications that any regulatory approach has to be on a case-by-case basis. And I think, for instance, the recommendation from the WHO and the document that Owen showed to us before, that's already included. It's a very good starting point for how gene drive can be used eventually in the field.
Yeah. Yes, thank you both. And unfortunately, we don't have much time left, but hopefully we have addressed some important aspects of the different types of gene drive and their relevance for policy development. Thank you all for this fruitful discussion. It was interesting to learn there are many different kinds of gene drives, like homing-based drives, split drives, Y drive, methanol toxin drive, and so on. And for the regular policy for gene drives, we must ensure that the trials are scientifically, politically, socially robust, and widely transparent. And we may even need to assess case by case. Now to finalize our event, I would like to invite Dr. Mahali Ahuja from ISAA to say a few words. Dr. Ahuja is the global coordinator of ISAA and the executive director of the Malaysia Biotechnology Information Center. He also serves as a public partition and outreach strategy development for the UNFAO Sri Lanka Biosafety Program, and as an adjunct lecturer at Monash University, Malaysia, and the AIMST University. Welcome, Dr. Ahuja.
Thank you so much, Dr. Wang. And before I close and before I say my word of thank you, I would like to call for the evaluation survey to be flashed on the screen. Or also please share it on the chat as well. We have so many participants asking for the certificates of attendance, and please remember, you only get your certificates if you fill in the evaluation form. And this is so important because this helps us to customize our future webinars for your need. Do we already have that?
Yes, it's already.
Okay, good. So please have a look at the evaluation link from the chat. And on behalf of ISA and Outreach Network for Gene Drive Research, I would like to thank all the speakers, both those speakers, excellent presentation and very well conveyed, to the moderator and to all the participants for this very engaging webinar. We have passed 20 minutes and this shows that this webinar is really well accepted, and also the demand and the need for webinars on Gene Drive. This actually marks our second year of webinar series on Gene Drive. Although Gene Drive is still considered novel, we are seeing great interest in this topic around the world based on our participation level, this webinar, and as well as when we did it last year. This is a good sign of proactiveness among technology developers to develop an enabling environment for Gene Drive to flourish, given all its benefits. ISA and Outreach Network for Gene Drive Research are being proactive in creating awareness among all stakeholders, so policies and regulations can be developed based on science. Dr. Edwards, you mentioned the importance of stakeholder consultation, and we hope our webinar series provides the foundation knowledge for such consultations in all your countries. Stay tuned for our upcoming webinars on Gene Drive, where we will be discussing risk assessment and risk management, and another one on integrating socio-economic and health aspects in decision-making. Let us all together put our actions together to build social licensing for Gene Drive. And also look out for our article on this webinar in the next Crop Biotech update. This is an e-newsletter from ISA. Please go to ISA website, isaaa.org, and you will see the website, you will see the newsletter there. You can subscribe to it. You will get it delivered to your inbox every Wednesday without fail. And on the website as well, you will also see a tab for webinars, ISA webinars. And under this tab, we will post both the presentations, and we will also have a link which will lead you to the YouTube, where the entire recording of this webinar will be there. So you can also watch it again and also forward the YouTube link to your colleagues so that we enhance the dissemination of information on Gene Drive. So with that, thank you so much for staying with us. See you in the next webinar.